Thank you for watching our video. This is Dr. K's Genetics Fall 2018 class. My name is Stephanie.
I'm Ann.
I'm Vincent.
And I'm William.
This experiment was an attempt to transform the smell of E. coli to a more pleasant banana smell by inducing it with a plasmid containing an ATF1 gene. This gene encodes for a transferase protein capable of changing isoamyl alcohol into isoamyl acetate, producing the banana smell.
So the reason we want to create the banana smell is because experimenting with these fundamentals of bioengineering can find applications and replacements for other industrial compounds such as paints, fertilizers, perfumes. So let's get started by learning a little bit about Igem and BioBricks and how we use them in our experiment.
Igem was created to facilitate the ability to insert and remove parts from a plasmid. The plasmid we desired contained a promoter, a ribosomal binding site, and the ATF1 gene. The plasmid containing the promoter and RBS we will refer to as Part A. To isolate this part, we use the restriction enzymes E. coli and SPE. The plasmid containing the ATF1 gene we isolated by using the restriction enzymes XBAL and PST. And to isolate the ampicillin backbone, we use the restriction enzymes E. coli and PST. After transformation of the E. coli, we placed it in an ampicillin plate to see if we got cell growth. As you can see, we got successful transformation by the colonies that were present.
Okay, so once we were able to see growth in our ampicillin plate, we used these colonies to create our desired banana smell by mixing our transformed bacteria with IPTG to induce the smell and isoamyl alcohol to be converted into isoamyl acetate. We then labeled this Tube 1. To test for the banana smell, we created two more control groups for different variables. Tube 2 contained more transformed bacteria with isoamyl alcohol, however, no IPTG, therefore no inducer to create the banana smell. Tube 3 was a mixture of untransformed bacteria with IPTG and isoamyl alcohol. We then conducted the sniffing test. The sniffing test consisted of having 7 subjects sniff our 3 tubes without knowing what's in the tubes, then answering a few questions and comparing the smells to a banana-flavored Laffy Taffy smell that we created by chopping up the Laffy Taffy and placing it in a 4th tube mixed with deionized water. In between tubes, we had the subjects sniff coffee beans in order to neutralize the smell. The questions we asked the subjects while sniffing were, what do you smell? How strong is the smell? And does the smell resemble the Laffy Taffy smell? All results were then recorded in this table. As we can see, Tube 1 had 5 out of the 7 subjects reporting that a sweet smell was present, if not the banana smell that we were looking for. And only 2 subjects reported bacteria smell. In Tube 2, all subjects reported bacteria smell. And in Tube 3, all subjects except for 1 reported bacteria smell. This 1 subject actually reported banana smell. So what these results tell us is that Tube 1, which was expected to smell like bananas, was successful in the transformation. Tube 2, since no IPTG was present to induce the banana smell, did not create the banana smell. And Tube 3, consisting of untransformed bacteria, also failed to convert the smell into banana smell. The one deviation in Tube 3 can be explained by the fact that when you mix IPTG with isoamyl alcohol, it does indeed create a sweeter scent. However, since the bacteria is not transformed to continue producing that smell, the smell will not linger. Some changes and extra questions that you might ask in a future experiment would be maybe having a bigger sample size and asking the subjects beforehand if they have any congestion or illnesses that might affect their smell. And also maybe having 2 rounds of sniffing or 3 and seeing if the subjects respond differently the 2nd or 3rd time around.
After cutting the pieces of DNA, we wanted to make sure that the restriction enzymes cut at the right places. In order to do so, we used electrophoresis on a small sample to run a gel that actually tells us the sizes of each of the digested pieces. We also ran this gel on uncut pieces to have something to compare them to. As you can see, our digested ampicillin-resistant backbone looks flawless, but there's a lower concentration of DNA in our Part A and Part B. This will affect the strength of the banana smell, explained later on in the results. While they were running the sniff test, we actually extracted some DNA from the newly transformed colonies and cut it at the EGO and PST1 sites. We did this to run a diagnostic gel, and we were hoping to find 2 pieces of DNA, 1 piece at around 2,114 base pairs for the ampicillin-resistant backbone, and another one at around 1,850 base pairs for the promoter, RBS, and the ATF1 gene. We also ran an uncut sample to have something, once again, to compare to. What we found that was really strange was that the uncut samples seemed to be shorter than the cut samples. So we ran it a couple more times to eliminate the factor of human error, but we kept getting the same results over and over. We then inferred that it was because the plasmid is circular, and so it has the capability of supercoiling, which allows it to travel faster through the gel than the linearized pieces of DNA. Something else we found that was really, really strange was that the linearized, supposedly complete parts of DNA with the expected sizes of 3,964 base pairs ended up being around 2,114 base pairs, which is the same size as the ampicillin-resistant backbone.
So one of the very interesting things that actually happened was that our plasmid is supposed to be around 4,000 base pairs, but when we only linearize the plasmid with one cut in either ECHO or PST, it turns out that the entire plasmid, or most of the plasmids, turned out to be around 2,000 base pairs. And because this bacteria with the plasmid is actually AMP resistant, we could infer or assume that it's somehow taken up a 2,000 base pair ampicillin backbone that somehow religated back together without taking any of the parts. Our results conclude that there are actually some bacteria that have taken up a fully constructed form of our plasmid. Due to the results of the gel, there doesn't seem to be a detectable amount of plasmid that were actually successfully taken in.
However, we could see that we had enough bacteria to produce the banana scent. We also noted that after 2 days of incubation, the banana scent was very minimal. And after 4 days of incubation, the banana smell was potent.
At this time, I think it's a good idea to address the questions that were asked during our presentation.
So, what is IPTG and how is this related to the LAC-I? Well, essentially, the LAC-I produces a repressor protein that prevents the transcription of the ATF1 gene. When introducing IPTG, which is very similar to allolactose, it actually binds to the repressor protein that's preventing the ATF1 transcription and promotes transcription.
We also noted that after 2 days of incubation, the banana scent was very minimal. And after 4 days of incubation, the banana smell was potent. So another question that was asked was, how did you deal with the satellite colonies? What we basically did was we found an isolated colony and we plated it on a different ampicillin plate to allow further growth. Another question we received was, why did we use ampicillin? Well, our part A and part B are in chloramphenicol backbone. We could have used other backbones like kanamycin and tetracycline, which we did, and our kanamycin backbone that we originally used did not grow any colonies. And so we changed to an ampicillin backbone that resulted in growth of the transformed bacteria. Some weaknesses that we found throughout our experiment was initially we did not have enough concentration of our parts. At this moment, we'd like to say a special thank you to Dr. Kirkpatrick, Professor Hines, and the entire biotech staff for helping us in every part of this experiment. We also want to take a moment to thank Igem for allowing us access to the parts that we desired. Thanks so much for watching our presentation. Yay!